Thank you, everybody. So today I'm going to talk about magnetic 3D cell culture and as a tool to bridge the gaps between 2D and 3D. So first, quick history of the technology, and that's probably why Caroline mentioned that I was in Texas. This is where the technology started. This technology, magnetic 3D cell culture started from collaboration between two big institutions, MD Anderson and Rice University. I was at MD Anderson. Other collaborators were at Rice. And we developed the technology. We partnered with Greiner early on. And then in 2018, Greiner acquired the technology from Nano 3D Biosciences. And that's why I'm Greiner. So quick motivation, why 3D cell culture, right? We know how to culture cells. And we also know that developing drugs is very expensive. And I like to give this angle because we always talk about that a lot of drugs when you're developing, they usually fail for poor efficacy or toxicity when they get to humans, right? So, and that's one of the reasons why it's so expensive to develop drugs. And part of the reason for that is because, we start testing drugs on in vitro. We're using monolayer cultures, which we call 2D cell culture. And as we know today, and I'll show some proof of that, monolayer cultures are not good representation of vivo biology. Therefore, it's no surprise that drugs fail when they get to humans. Then usually after drugs goes from 2D cell culture in vitro, they go to animals. And of course, animals don't have human physiology. So that also contributes to the high failure rate when it gets to human. So the idea here is to have 3D cell culture where you use human cells. You can generate an environment where cells grow like they do in vivo. So we can bridge the gap between the in vitro and the in vivo world and hopefully reduce the number of failures for poor efficacy and toxicity and make the drug discovery process cheaper. Now, a more practical view of 3D cell culture is when you go from monolayer culture for culturing cells in 2D to culture cells in 3D, you realize how important cells stuck to plastic is helpful when culturing cells. If you want to change media, why is it easy to change media? Because cells are stuck to the bottom. Immunofluorescence, immunohistochemistry. Why is it easy to do immunohistochemistry cells? Because cells are attached to the bottom. So when you go from 2D to 3D, you lose the cell attachment. And very simple tasks that are done in 2D are no longer easy. So our idea here is to use magnetic field as a surrogate for cells attached to plastic to make cell culture easier. Okay, so how do we do that? So you have your cells, and we add the nano shuttle. So why the nano shuttle? Because cells are now magnetic. We have to magnetize the cells. And the nano shuttle is a nanoparticle assembly of iron oxide, gold, and polylysine. Polylysine provides the interaction between the nanoparticle and the cell, and that's the cells just electrostatically interact attached to the cell. Then you use the same protocols you use with monolayer culture and detaching, counting, and you can place the magnetic field on top and actually make the cells levitate, or you can put the magnetic field on the bottom and bioprint the cells on a cell repellent surface. So those are the two approaches we have. And the levitation system we have in 624 wells. The bioprinting we have virtually from 6 all the way to 1,536. So this is just a quick tour of some of our results. Because we use magnetic field, and I'll get into detail, because the magnetic field you can use the cells, bring the cells together, and form the 3D structure very fast. Is the background me or somebody else is not on mute? Just me. Okay. Thank you. Appreciate it.
Now it's gone. Yeah, I muted the person. Yeah.
All right. Thank you. So you can use the magnetic field to bring the cells together and make spheroids very fast. You can co-culture cells, which is not trivial. And here on the top right, you have we have glioblastoma GFP labeled. The red is astrocytes, and we cultured them separately, and then we magnetically brought them together. And you can actually see the green, the glioblastoma invade the astrocytes. And on the bottom right here is an adiposphere, is a co-culture endothelial cells and pre-adipocyte stem cells. And couple of things I like to mention about this result here on the bottom right. First, this was the cover. Oh, sorry, I went too fast here. 1 second. First, this was the cover of Tissue Engineer a few years ago, and the reason for that is because we were able to stain this tissue whole mount. And what I mean by that is you can use the magnet to hold the spheroid on the bottom of the plate. And you can stain just like you do with monolayer culture. And that's, if you've done 3D cell culture, that's not trivial. So we can really do all the manipulation and come up with this beautiful image. And also, this was a co-culture of pre-adipocyte stem cells and endothelial cells, where we were trying to replicate the endothelial cell phenotype within the culture. And when we first started, we used 50-50 ratio of cells, 50% of pre-adipocyte stem cells, 50% of endothelial cells, mix them together and observe, see if we got this sprouting of the cells within the culture, and we didn't see any. For us to actually see the endothelial phenotype, we had to titrate the culture down, the number of cells down from 95% of pre-adipocyte stem cells, 5% of the endothelial cells. And that's very important because when you go to 3D cell culture, more complex environments, you want to co-culture different cell types, you're going to have to optimize to achieve your the desired phenotype. And that's what we can do with magnetic 3D cell culture. We can really help the manipulation and optimize your experiment and the way you want. So this slide I like to show for a few different reasons. One reason is that I don't know why it's probably timed, but that's okay. So the best marketing is a good publication. So we, as a small company, when we moved from MD Anderson to Nano 3D Bioscience, worked really hard to publish very good papers. And as you can see, we have published really well. And the next reason for it, which comes to this slide, is that, which is a fair question, is the nano shuttle biocompatible? And it is. It has no effect on the cells. And we've shown over and over on these publications. The reviewers ask, or used to ask, what the nano shuttle does to the cells. And we have a 5-page white paper that shows viability, proliferation, with and without the nano shuttle, protein expression, stem cell differentiation, inflammatory stress, oxidative stress, gene expression, and there's no difference on cells with and without the nano shuttle. So it's biocompatible. And as I said, if you have any questions, just let us know, and we can send you the results and the white paper that compile all the results. So the system is very simple. Basically, it's a magnet sitting right in the middle of the well. And you magnetize your cells, you put in the well, and the cells go towards the magnet, as you can see here. And then with most cell types, with the cell lines, you only need to leave on the magnet for 15 minutes. You take the magnet off, and cell-cell interaction take over and makes the spheroid. So here's a quick movie that shows the exactly workflow of the technique. So you have your magnets, and this is a kit. So you have your magnets. So first the nano shuttle, you mix the nano shuttle, add it to your cells. It's just incubate overnight. The nanoparticle is attached to the cells. You wash your cells the next day, and the free nano shuttle comes off. You detach, you count your cells, And then you take your cell repellent plate and you point what we call the spheroid drive, the small magnet that sits right in the middle of the well, and you dispense your cells. So as soon as the cells see the magnet, they come together. 15 minutes on the magnet, and you can see the cells come together. Again, this is a flat surface, and cell-cell interaction overnight takes over and makes the spheroid. All this done on a flat surface. So now you want to change your media, you want to add your compound, you want to have fresh media. You put it on the larger magnet to hold your spheroid down tight. You replace your media, and you come and you add your compound. So that's the, you know, a drug testing workflow, and that's it. So, and here you see 96, 384, and 1536. And as we say, it's a 3D and a 2D workflow. And of course, reproducibility is one thing that it works really, that it's a hallmark of the system. This is actually a application note that we did with Promega a few years ago, just showing that how reproducible the system is using 384 well plate system. From 600 cells all the way to 20,000 cells, you have cell number and you have size of the spheroids. The error bars here are the standard deviation, so very tight size distribution. And on the bottom is your cell number versus chemiluminescence signal from cell titer glow. And you can see a very linear curve for a wide range of cell numbers. So the first set of results I want to show quickly here is we're using cardiomyocytes. These are iPSC-derived sites that we magnetically bioprinted them in our 384. And you can see there's whoever works with cardiomyocytes, you know, really enjoy it because it's great to see the cells beating. And you can see these cells beat for actually over 19 days. And this is on the right here is just a plot of the beating at day 7. And on the same topic, in illustrating the enabling aspect of the technology, this is work done by AstraZeneca in the UK. And as you know, measuring electrophysiology, you know, patch clamp is not the easiest thing in the world. But when you go to 3D cell culture, it's really hard. So measuring electrophysiology, it's not trivial with different cells. So what we did here, what AstraZeneca did here, they used the magnets actually to position the cells on the electrode so they could measure the electrophysiology of cardiomyocytes. And here are the results. So they magnetized the cells, they used the magnet, and they were able to make the spheroid to sit right on the top of the electrode. And then they used impedance field potential measurements, and you can see the left plot is I'm just gonna go off here and come back because my pointer is not working. So so you have impedance field potential on the left is on the magnet. You can see very reproducible beating signal. And if you don't have the magnet, it's just noise. It doesn't really work. So this was a really cool work that they did. And again, and I believe they used the NAN-ion system where they guided the spheroids to the electrodes. So one tool that also application that we're developing is also making multiple spheroids in the same well, which is something that people are interested for different applications, for expanding cells. We have some customers that use this to expand patient samples in the same well. So here you have a picture of a 6 well where we have several spheroids. And from this picture, you can see they're fairly the same size. And we're just varying the number of cells here, 20,000, 10,000, 5,000, all the way to 600 cells per well. And the way we accomplish this is very simple. Basically, we use we call the magnet drive, the array of magnets, 384 magnets, and we place a 6-well cell repellent plate on top of the magnets, and you get about 50 spheroids per well. So you can get very reproducible spheroids, 20,000 per spheroids, 10,000 all the way to 600 cells per spheroid. And these are pictures of spheroids in the same well. So, and again, this is not trivial to do in 3D. So we're just using the magnetic field to really enable the manipulation and control of the positioning of the cells. So next question, the next topic, and I started with a question because we often get the question, how can we easily transfer the spheroids from one plate to another? So again, my question to you is, could a simple tool to transfer spheroids help you? And we get this question. People come and say, you know, I cut the end tip of my, the tip of my pipettes to transfer, don't break the spheroids. So what we came up with is what we call the multi-mag pen system, which we have in 20. Actually, we have a single 24 and 96. The new is the 24 and 96. And here's how the system works. So you really have a 96 well plate, and you can see the spheroids attached to this plastic lid, which has a magnet inside. And you can very easily transfer the spheroids. And the exact protocol or process, so you have your plate here, and you have a spheroid in the middle of the plate, as you can see. Then you have a plastic sleeve, which looks very much like a PCR plate, but it's not, because we modify this plate so the cells won't stick to it. And then you put this this plate inside of your cell 96 well plate, this sleeve. Then you put a magnet inside of the sleeve. So the magnet's never in touch with the cells. And then you're able to pick up your spheroids very easily. Now, how do you transfer now? Then you take your, you remove the magnet from the plate. So your sleeve is there still, and then you put, actually first, one step I didn't miss. And here you go to a new plate, right? So you take the magnet off of the old plate, you insert this in the new plate. And with the magnet on the bottom, you actually are able to pull the spheroids off the plate. And as you see, all the spheroids were transferred. So we started with here with the spheroids on the magnet, and you transfer all of them to a new plate. So again, that's very nice tool, and you can very gently pick up and transfer the spheroids. And we also have this in a 24 well format. This will be available in January. And if you have any questions, just let us know. Next, of course, if you're talking about 3D cell culture, cancer applications, right? And I'm going to follow the theme of the enabling tool of magnetizing cells, because here we're culturing NALM6 cell line. And NALM6 cell line is a leukemia cell line. And that means that the cells are blood cells, and they don't form 3D structures. They're circulating cells. So why are we using 3D cell culture with a cell that doesn't generate 3D structures? We're using the magnetic 3D cell cultures to make it easier to manipulate the cells. So the goal here was to measure the specific targeting of CAR T cells on CD19 positive leukemia cells. So they're looking at CAR T immune cell, CAR T targeting. So instead of using the chromium assay, which is traditional assay, which is nasty, we actually took CD19 positive cells, and they express red fluorescent proteins, and we took a CD19 negative, which express a green fluorescent protein. And we mix the two together. We magnetize them, and then we mix the two together. And with the magnetic field, we focus them right in the middle of the well, okay? Then we added the CD19 targeting CAR T, which is not magnetized. And after 48 hours, watch what happens. The red cells, the CD19 positive cells, are gone, or mostly gone. If you really zoom in, there's still some left. But you could specifically see the targeting of the CAR T by just focusing the cells in the middle. So this is just enabling tool. By magnetizing the cells, we can use, we can wash these cells, we can manipulate, move them, and we can detect the CAR T activity. Next, going more to the, close to home 3D cell culture traditional approach that we're looking at. It's traditional, but it's not that traditional because this is actually a PDX comparison we have from our system with PDX. So here was a collaboration with Jeff Bartholomew at MD Anderson. We took patient PDX cells. Actually, we took patient samples, triple negative inflammatory breast cancer. We culture in the animal, and then we took the tumor and we compared those same cells. If we take those cells and make a 3D culture, how does it compare to the original tumor? So here's the work. So we took the tumor, patient to animal, animal to here. We split the tumor in half. Half we used the traditional methodology, pathology methodology that they have in the lab. The other half, we came up with a protocol which we broke up the tumor. We magnetized the cells and we reassembled the culture magnetically. And here are the results. So we look at ECADIA expression here, Vimentin, and Ki67, Ipismag. So the left is the tumor, the right is the culture, and this is a blown up picture of those results. So tumor on the left and two different bioprinted cultures in the middle and on the right. So, and this is immunohistochemistry staining, is a paraffin embedded and sectioned, and you can see the marker distribution comparison between the 3D culture and the tumor is exactly the same, very close. And in this paper, we also showed the gene expression, which is the same, and drug activity. They are also correlated to the in vivo results. So this paper we published a couple of years ago. And if you have any questions, let me know. And one question I do get from when I show this data, or when I show data in general, because this is the ultimate comparison, is the in vivo comparison to 3D. Very close resemblance. And people ask me, so do you have the 2D comparison? In this case, these cells did not grow in 2D. So we couldn't even, Jeff could not grow the cells in 2D. So this is the only way we could really do was 3D. And if you're curious or want to know more details on the results, let me know, because I helped them develop this method. So we'll be glad to help you if you're working with patient samples or PDX samples. And here is the results of patient samples. This was collaboration also in Houston with the University of Texas Health Science Center in Houston. And we took a small biopsy. This was a bone biopsy of a prostate cancer metastatic site. From that small sample, we broke it apart, we magnetized it, reassembled, and in 72 hours from the biopsy, we're able to show a dose dependence curve for doxorubicin. And of course, this tumor is supposed to be EpCAM positive, and it was EpCAM positive. We put it back on the magnet, and we stained. So this is really moving the application of the magnetic 3D cell culture towards personalized medicine, where we can really handle small model samples, and we can really miniaturize the cultures, if you may say, by using the magnetic field and compressing the cells together. And last, but not least, this is close to my heart, and I like to show this is the space station, and we did do an experiment, a project in the space station. And again, following the same topic of enabling tool. This is, I don't know if you know, but the reason we are able to culture cells in 2D is because there's gravity, right? Gravity pulls the cells down. And when you go to space, cells grow in 3D. So you don't really need a 3D cell culture tool in space. So why are we doing a project in space with a 3D cell culture technology? The reason is because in space, changing media, the same things I was talking about, manipulating the cells, changing media, staining, it's very hard. And so in a way that the way we approach this is actually to send cryopreserved magnetized cells in space where we could use the magnetic field to actually hold the cells down and manipulate them, change media, and it worked great. And of course, this is Peg Whitson. That's her work with our system in the space station. And right after she came back, she was on National Geographic cover. And one more thing that is from the science point of view, one thing that we tried to do in space was to actually create monolayer control in space, right? Because cells only grow in 3D in space. So we wanted to have a membrane where we could put the cells on a surface and grow them in a monolayer. And as anybody that has done an experiment in space, it's or in general, just experiments, the results didn't come out as planned because as expected, because we were able to attach the cells to a membrane, but they never really spread and grew in 2D. They actually, they kind of grew as in a 3D structures with some cells attached to the membrane. So we have more ideas and more experiments to do to our next trip to space. But again, I just wanted to highlight the enabling tool of using the magnetic field, that it's really widely applicable. And of course, this is a large amount of work. We have lots of collaborators, and I have to acknowledge them. As you can see, this is there's a bit of flavor in the presentation because this is it's a lot of research with a lot of great researchers. And if you have any questions, please.
Yeah, thanks a lot, Dr. Sousa, for this very fascinating talk and this enthusiastic presentation of your data. So now would be the possibility for everyone to ask a question, either via raising the hand or via maybe using the chat function. This would be possible now. Do I see any raised hands? Not yet. Okay, so if it is not the case, did you try it on neurons? Is one question.
We have tried in neurons. Sorry, I didn't show, but I'll show you quickly since we're all here. I can just bring it up. Again, 30 minutes. It's we have lots of results. We have worked with neurons. This is work we showed at neuroscience a couple of years ago in collaboration with BrainCell and biotech. So here the greens are the neurons, the red are smooth muscle. And we actually saw the neurons line up within the culture. And we do have the poster on our website. If you send your email, we can send you the poster, and I'll be glad to show you details of how we did this work.
Perfect. Also, thank you for the opportunity and the offer. There's also asking someone if the slides will be available. I would say if no one of the speaker has any doubts, then we can make the slides for sure available on our LSR VDGH homepage. I can also show, there's also another question. Regarding the cell function, like secretion of components, for example, do you see a difference between 3D and 2D cell culture?
Yes, we've seen difference on different proteins. Of course, some proteins are the expression is the same, but we certainly do see difference on excreted proteins between 2D and 3D.
Okay, perfect. Thank
So
you.
of course, that would depend on the specific proteins, but yes, we have seen in different markers. Yeah.